Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET entrance exam question paper. This is PG entrance that I will be discussing. It was conducted in the year 6th June 2023 for the subject Life Science. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CUET MSc entrance exam, then you can go to my channel playlist. That is Omni Bio Entrance is the name of the channel. Go to the playlist, see for CUET. In that you will find the previous year question papers. Coming to the first question. Which radioisotope is used to label the 5' end of DNA or RNA? It is S35. No, DNA doesn't have sulfur. Then phosphorus, P32. Yes, DNA and RNA both have phosphorus in their backbone. So you can use P32 for labeling. DNA or RNA. C14 no, and C12 also not used. Two-dimensional electrophoresis resolve the proteins on the basis of. So two-dimensional, it resolves the protein based on two dimensions. The first dimension is based on isoelectric focusing and separates protein according to their isoelectric point. In the second dimension, you use SDS page and it separates protein according to their molecular weight. So two-dimensional electrophoresis resolves protein on the basis of their net charge and mass. Answer is option 3. Then next one. Differences in the absorption of left and right-handed circularly polarized light is measured by. So it is measured by CD spectroscopy or circular dichroism spectroscopy. Answer is option 3. Then next one. The radiation source in TEM, that is transmission electron microscopy, is. So in transmission electron microscopy, you use electron beam as the radiation source. Answer is option 2. Full form of MALDI MS is. So MALDI MS is matrix assisted laser desorption ionization mass spectrometry. Answer is option 4. Then next one. The most distinctive feature of phylum Echinodermata is the presence of water vascular system which helps in. So water vascular system, it helps in locomotion, capture and transport of food and respiration. So all three are given in option 4. So your answer is option 4. Then next one. The hormone which stimulates an appetite is. So out of these it is ghrelin, and that is option 3. They have made a printing mistake. It is GHRELIN. Answer is option 3. Then next one. Name the Nobel awardee who provided the first direct evidence that different genes are physically located on the same chromosome by X-linked pattern of inheritance in Drosophila. So in Drosophila it was T.H. Morgan. Answer is option 2. What is the end product in the process of glycolysis in anaerobic and aerobic condition respectively? So under anaerobic condition you get either alcohol or lactate, and under aerobic condition you get pyruvate. So alcohol is not given in the option, it is lactate and pyruvate. Answer is option 3. Then next one. Which of the following secretes progesterone hormone? So it is the corpus luteum that secretes progesterone hormone. Answer is option 2. Two envelope spikes in influenza virus, HA and NA stand for. So in influenza virus, HA stand for hemagglutinin and NA stand for neuraminidase. Here also for neuraminidase they have written L instead of R. And that is a printing mistake. So answer is option 2. The polar head groups in sphingomyelins is composed of. So sphingomyelins, the polar head group is composed of phosphocholine and phosphoethanolamine. Answer is option 3. Then next one. Which of the following statement is incorrect about gel electrophoresis? So it can be gel electrophoresis. They have given a general term. It can be either protein or DNA. So first one. In proteins or nucleic acid fragments are separated based on their net charge and mass. That's correct. Differential migration of these molecules, that is either your protein or nucleic acid, through agarose gel or polyacrylamide gel in which small particles move slowly to resolve better. This statement, that is small particles move slowly, that is incorrect. In both agarose and polyacrylamide gel electrophoresis, small particles actually move faster, not slower. So they have asked for the incorrect statement. So 2 is correct answer. Then electrical charge that is on DNA molecule help them to move towards the anode. That's correct. Because DNA is negatively charged because of the presence of phosphorus backbone. So it will move towards the anode. Then migration of small DNA fragment through gel is quickly. That is also correct. Then in bryophytes, next one. Sporophyte is partially or fully dependent on gametophyte. That itself is correct. Xylem and phloem are the main water and food conducting tissues. So by bryophytes, as you all know, they are non-vascular plants. So xylem and phloem are not present in bryophytes. Spore dispersal take place by means of peristomial teeth only. Not only peristomial teeth, there are elaters, wind and water through are the means by which spore dispersal take place in bryophytes. Heterospory and seed habit evolved in bryophytes. Heterospory seed habit they evolved in pteridophyta, not bryophytes. So option 1 is the right answer. Then next one. Self-incompatibility prevents self-fertilization and this mechanism involves. So they have mentioned in the question itself that self-incompatibility is mainly used to prevent self-fertilization. So if you see option 1, S1 pollen grain from S1, S2 parental sporophyte cannot fertilize eggs of an S1, S2 flower. Yes, that is correct because pollen grain is S1 and eggs are also from S1. So S1 and S1 are both present in pollen grains and eggs. So it will not fertilize. That is correct. This is because the S1 pollen grain, it carries the same allele as the female plant, that is S1 and S2. So this recognition, it triggers a self-incompatibility response. And thereby it will prevent the pollen grain from germination on the stigma of S1, S2 flower. So first answer is correct. Then next one, if you see S1 pollen grain from S1, S2 parental sporophytes can fertilize eggs of S1, S2 flower. No, they cannot fertilize. S2 pollen grains can fertilize eggs of S1, S2 and S2, S3 flower. No, S2 is present in the egg and S2 is also present in the flower. So S2 pollen grain cannot fertilize. S1 pollen grains from S1, S2 parental sporophytes can fertilize eggs of an S1, S3 flower. No. S1 is present in the egg also. So S1 pollen grain cannot fertilize. Then next one. What is the composition of membrane attack complex in complement system? So in complement system, in the end they will form what is called as a membrane attack complex after all the complement pathways. This membrane attack complex composition they have asked. So it is C5b, C6, C7, C8 and C9. Answer is option 4. Which molecule ATP does not donate? So ATP structure, it has got an adenine, that is the base, a ribose sugar and 3 phosphate groups, that is alpha, beta and gamma. And ATP donates phosphoryl groups, that is gamma phosphate, adenyl groups, that is AMP during ATP hydrolysis, and deoxyribosyl group, a component of it is a component of the DNA sugar phosphate backbone, so it will not be able to donate deoxyribosyl group. So which does not donate, they have asked. 1 also is correct, but 2 is more correct. Deoxyribosyl group is more correct. Phosphoride groups, it is not present. Phosphoryl groups and adenyl groups ATP donates. Then next one. Which microorganism produces dextran? So it is leuconostoc mesenteloids. Answer is option 2. Then next one. In chloroplast, ATP generation by chemoosmosis mechanism involves the diffusion of hydrogen ions from stroma to stroma in the presence of light. No. It is, if you see the photo here, it is diffusion of hydrogen ions from the thylakoid lumen to the stroma of the chloroplast. It results in the formation of ATP from ADP. So the answer here is, if you see option 4, the diffusion of hydrogen ions from the thylakoid space back to the stroma through ATP synthase complexes. Answer is option 4. Then next one. Which of the following is correct about RNA interference? So RNAi take place in all prokaryotic organisms as a method of cellular defense. That is wrong. RNAi, it is primarily found in the eukaryotic organism, that is animal, plants, fungi and protists. It is not found in any prokaryotic organism. And prokaryotic organism, they have an entirely different mechanism for cellular defense. That is CRISPR-Cas system, small RNA mediated regulations, ribose switches, RNA sequence based regulation. So first statement is not correct. Second one. RNAi involves silencing of a specific mRNA due to the complementary double-stranded RNA molecule that bind to and prevent translation of the mRNA. That is correct. So you have got a specific mRNA which you want to silence. If you create a sequence complementary to that, this double-stranded RNA, it will prevent the translation of the specific mRNA. Third one, if you see, RNAi involves silencing all types of RNA. That is not correct. It is specific to mRNA. RNAi involves silencing of a specific mRNA by the host cell itself due to the overproduction of mRNA that is not correct. Then next one. The precursor molecule for the synthesis of vitamin D3 in human skin is 7-dehydrocholesterol. Answer is option 2. In photoperiodism, long day plants flower only if night is longer than critical dark period. Okay. So what is photoperiodism? It is the response of plants to day length and it influences flowering. So long day plants means they will flower only if day length is longer than the critical day length or night length is shorter than the critical dark period. Only then long day plants will flower. So the answer here is option 2. If you see, they have mentioned if the night is shorter than the critical dark period. So option 2 is correct. A flash of light interrupting the dark period prevents flowering. Actually, it promotes flowering in long day plants. So 3 is not correct. Then fourth one, a light period of less than 8 hours is sufficient for the plant to flower. No. Long day plants, they typically require light period longer than 12 to 14 hours and night period shorter than 10 to 12 hours for flowering. So fourth option is also not correct. Then genesiogamy is a type of pollination in which. So it is a type of self-pollination, and we will see what genesiogamy is. It is transfer of pollen grains from anther to stigma of another flower of the same plant. That is called as genesiogamy. Answer is option 3. Then citronella oil is obtained from. It is obtained from Symphopogon nordus. Answer is option 3. Then which is not the heteropolysaccharide of the extracellular matrix in multicellular animals? So which is not the heteropolysaccharide, you have to see. So hyalurotanine, keratin, sulfate, heparin, they are all heteropolysaccharide, but keratin is not a heteropolysaccharide, it is a structural protein. So the answer here is option 3. The next one. Which of the following is one of the possible hazards of genetically modified crops? Modified genes of herbicide resistance from GM crops get into weed plants through pollination. That is correct. Herbicide resistance gene which you have introduced into a GM crop, it can get transferred to a weed growing nearby the GM crop through pollination. And this plant might become resistant to herbicide. That is also correct. And so becomes a superweed. That is correct. So first option is correct. All the other options, that is there is no possible hazard known till date, is wrong. Production techniques of GM crop is proof, and genetic engineering offers the advantage of transferring on these genes that are required. No. Production of GM crops may replace the existing germplasm. That is also not correct, but more correct is option 1. A nerve impulse is transmitted from one neuron to another through a junction called synapse. Everybody knows that. Answer is option 2. Then application of genetic fingerprinting is not directly linked with. So identification of suspects, yes. Determination of paternity, yes. Species conservation, yes. That is identifying species, tracking populations, and understanding genetic diversity. In all these cases, you use genetic fingerprinting in species conservation. Property disputes, no. Genetic fingerprinting doesn't have anything to do with property disputes. Answer is option. Then next one. Extracellular genomes of mitochondria and chloroplast have evolved from. So they have evolved through a method called as endosymbiosis. Answer is option 4. If the template strand of a segment of a gene has the nucleotide sequence GCTAAGC 5', what nucleotide sequence will be present in the RNA transcript? That is your mRNA specified by this gene segment. So the segment is in the 3', 5' direction. So you have to see for the mRNA transcript in the 5', 3' direction, and it will be complementary to this sequence, except that instead of T, U will be present because it is an RNA sequence. So if you see that, if you see option 2, you can see that CGAUUCG is the correct answer. Even if you don't know the complementary sequence, if you see the other options, everywhere it is T is present, not U is present. So those 3 options are not correct. Then J chain is present in. So J chain is present in IgA. Then Hardy-Weinberg equation can be used to test whether evolution is occurring at a population level. Yes, that itself is correct. Then in interface cell less compacted and highly condensed chromatin are called as. Okay, less compacted are called as euchromatin and highly condensed chromatin is heterochromatin. So the answer here is option 3. Which is not the antimicrobial peptide? So antimicrobial peptide, it is immunoglobulin. Answer is option 3. The function of telomeres is, so telomeres, they are present at the ends of the chromosomes. So they protect intact eukaryotic chromosome from improper fusion or degradation. That's correct. Attack the intact eukaryotic chromosome at the centromeric region. No. Degrade the intact eukaryotic. No, they don't degrade it, but they protect it from degradation. The ends of the eukaryotic chromosome. Protect the intact chromosome from histone proteins. No. That is also not correct. The karyotype of individual in written as 47XX plus 21 indicating. So plus 21 in trisomy of the 21st chromosome. That is why there are 47 pairs, 47 numbers. So it is Down's syndrome. Answer is option 2. Lamb-Rush chromosome were first discovered in salamander oocytes by Walter Fleming. Answer is option 4. Then topoisomerase 2 introduces the, so topoisomerase 2, they introduce negative supercoils in DNA. Answer is option 3. The sequence essential for centromere function are, so they are about 130 base pair long and are rich in AT pairs. Answer is option 3. The sea anemones and corals belong to the phylum Coelenterata. Answer is option